Hi. This is problem solving session 5, and in this session, we're going to discuss the topics that we have gone over this week. So, basically, today I will be speaking about three processes which all come under this central term called central dogma of life. So, we'll start with this, we'll see how these processes are related to one another, and then we'll see each of these processes individually. So, central dogma of life is given by Francis Crick, and he mentioned one line saying that the central dogma of molecular biology deals with the detailed residue by residue transfer of sequential information. It states that such information cannot be transferred back from protein to either protein or nucleic acid. So there's a directionality to the flow of information in the cell or in the molecular biology. What he meant was the end product is protein, and there is no back information or backflow of information from protein to the nucleic acid. And how do you picturize that? You picturize something like this. So you have three processes: replication, transcription, and translation. And you have three macromolecules, three biomolecules that are important for your cells functioning. You have DNA, then you have RNA, and then you have proteins. So how these molecules are related to one another? So if a DNA is synthesized based on DNA, it is called replication. This DNA has all the information that one needs to be coding for protein. Proteins are the end product which will carry out all the important functions as we had gone over in the previous sessions. So this DNA will be transcribed to RNA basically. So this DNA will now be taken into an RNA form where RNA sequence which is complementary to the DNA will be synthesized. And this process is called transcription. And then you have RNA, which has all the information necessary for a protein formation, will be translated to protein by ribosomes. So these proteins will then be used for all the important functionalities in your cell. So that's how these three processes are connected: replication, transcription, and translation. But there are some processes which deviate from the central dogma. As I had said, there can't be a backflow of information from the protein to DNA, but several studies on different systems have found that this can also be violated. So first thing is reverse transcription. So as the name says, what was transcription earlier? Transcription was DNA to RNA. So what would reverse mean? RNA to DNA. So RNA will giving rise to DNA. So where does this happen? It happens in few viruses. So in those which contain RNA as their genomic content, and there the RNA is first converted to DNA. That is then used for replication. So you need first there to be a DNA, with then after the DNA, normal processes will be followed for transcription and translation. But the first step of getting to the DNA is by reverse transcribing the RNA to DNA. That's the first deviation. The second deviation is again RNA replication. So you might not need even to go to the DNA form. You just basically start with your RNA, but your RNA first has to be duplicated, as we had seen in the previous slides. So RNA will be duplicated, and then that will be used for synthesis of protein. And how is that done? That is done by something called RNA dependent RNA polymerase. So RNA polymerase means synthesizing RNA from RNA itself. Two other processes also deviate from central dogma. The first is DNA directly to protein. So you skip the RNA part completely. You directly go from DNA to protein, and this is possible in some in vitro cells. So when you do protein expression in vitro, you insert your gene of interest, that is a DNA, and you get the end product as the protein. So you don't know if there is an RNA step in between or not. So that's DNA being directly translated to protein. And the last in protein, again, these are in some prions. So you have a normal protein which is this form, and that will be itself giving rise to the disease causing form of the protein. So this also happens when you simply go from one protein to another and you don't have any other of these macromolecules included. So that's the central dogma. And we'll start with first process, which is a major process, which is a DNA replication. So very easy. This is something that you will find in my previous presentations as well. So this is DNA in its native form, and this is the complementary strand to this DNA, where you have at each position the base which is complementary to the respective position in the other strand. So let's say this is A, so the complementary base in the new strand will be T, and that's how it proceeds. So DNA replication refers to making multiple copies of the DNA strand by polymerization reaction, and this is carried by DNA polymerase enzymes. And there are several steps for these replication. So any process that we are going to read today, replication or transcription or translation, have a set of three steps which are always in the sequential order, which are initiation, to initiate that process, elongate, to elongate the process, basically to first of all start transcribing or translating or replicating. Elongate means to get to the total length or whatever the mechanism is, and termination is to stop that process. So initiation, elongation, termination is constant across all these different processes. And for DNA replication, you would need DNTP, and you would need a primer template junction. What that means is that to synthesize a new strand, you will need some DNTPs that could be added complementarily. So that DNTP is requirement for DNA replication, and you will need a primer template junction. We'll come to what is primer and why is it needed, but you will need this junction which gives this 3' OH end, so that you can continue your synthesis. Um, Okay, so if you have to write an equation for this DNA replication, how does each step proceed? So you will have a XTP. XTP means certain number or certain DNTP that will be joined by that will be joined to this XMPN. So some N, basically what you mean, let's say there are two nucleotides, and on that you will use one trinucleotide, and then it will finally become 3 nucleotide, and you will release 2 phosphate. So every DNA replication which involves an incorporation of a base, there is a subsequent release of 2 inorganic phosphates, and we'll see all of this in detail in a while. Okay, so we'll come to DNA replication steps, where I will start with initiation, elongation, and termination. Now, more or less all these steps are common in the prokaryotic system and the eukaryotic system. However, there are finer, minute details, and that's because how these cells are structured, how the nucleus are structured, how the genomic material is structured. But the overall idea is same for both eukaryotes and prokaryotes. But what you need to remember is the name, and there are some specific processes, let's say for, if you will see in a few moments, for initiation of DNA replication in eukaryotes. So those are very specific, but for some, you can just remember the overall flow, and that would be the same in both the systems. Okay, so let's start with first initiation. Now, what is initiation? What do you mean by initiation? Initiation means to start a DNA replication. So now you have a sequence, and if you randomly start replicating from anywhere, or any part of that DNA starts to be synthesized, it is not a good idea, because first of all, DNA replication has to be very accurate. You need to have the complete set of information transferred to the next new strand that is being synthesized. And secondly, if there are inaccuracies, it can lead to many problems. For example, if there are wrong bases incorporated, or there are some segments which are not at all replicated, there is loss of genomic content in the new strand, which is not what the cell wants. So cell has a very, or replication in a very, very regulated way, and it has certain sites from which this initiation starts. So um, so as you can see by the name, that origin of replication is where you will see the replication to begin. So now imagine this is a linear strand, and they are all bonded to each other. For any DNA to be replicated, there has to be first this breakage of these bonds between the original strand, so that you get this kind of nascent single strands. And there will be the space, then there would be the new strand synthesis. Basically what I mean is, if you break off this bond between these bases, then you have something like this. The DNA opens up because the bases move apart from each other, so the DNA strands move apart from each other, and now you have spaces in between where you can have the new strand synthesis. So where does this opening happens? Those sites are called origin of replication. And when you have this opening of the DNA strand, they are called the replication bubble, because you can see these are some circular like structure where you have open space in between. So that's why it's called replication bubble. So this origin of replication, as you can see, if it is a linear chain, or if you imagine the chain to be a long stretch of base pairs, there are several origin of replication that can open up and give rise to these multiple bubbles. So now this is important, because let's say you have E. coli. And E. coli had just one origin of replication in the entire genome, but it is also good enough so that the entire genome is copied within a certain limit of time. Do you know how long E. coli takes to replicate? It takes 20 minutes. So its genome is well structured so that even with one origin of replication, it can cover the entire genome in 20 minutes, and then not just replicate your DNA, but replicating the DNA and packaging the DNA into two daughter cells. That's also happening in 20 minutes. But for some organisms which have the big genome content, one origin of replication opening might not lead to complete synthesis of the entire genome in the given time. So then you can see such kind of multiple origin of replication and multiple opening. So what will happen if you imagine all these will be replicating in the simultaneously, and you have the entire genome being replicated in a certain given amount of time. All right. So this is how you see. So if you just have to see the figure, so if the origin, this is a replication bubble, and as and when the strands are getting polymerized, you have the strands increasing in length. Just a minute. Yeah, all right. So that's what I was saying about origin of replication, and it follows by unwinding of DNA at the site for these positions in the genome. Okay. Now, these origin of replication are called with certain name, and you will see how elaborately people have studied to see how this phenomena happens. So in E. coli, this origin of replication is called ORIC. And now imagine if you have to open up your DNA and you know that there are two kind of bond, AT bonds and GC bonds. Which of these bonds is easier to break? The one with two double bonds, two hydrogen bonds, or three hydrogen bonds? Of course, two hydrogen bonds, because that will require lesser energy to break them apart. So that's why your ORIC region, which is the region where this DNA will unwind, are AT rich regions. And for opening up of DNA before any replication can happen, there are several proteins that come and bind to these locations, so that once they bind, they can use up energy. Now, how are these energy? Let's say ATP or GTP. So they will hydrolyze ATP, GTP, get some energy, and that energy will be enough to break these bonds apart. So we'll see how that happens. So let's say this is the ORIC. Now you have a protein which is called DNA-A. So DNA-A is bound to ATP. Now DNA-A binds to certain regions in ORIC, and those regions are, let's say, 9 mers, 13 mers. What do you mean by 9 mers? Set of 9 nucleotides being repeated with one another. So you have 9 mers, 13 mers, and 5 mers. So this ORIC will be consisting of these kind of sequences. So these are all base pairs. You have 9 mers repeated 5 times, you have 13 mers repeated 3 times, and you have 5 mers. So you see, these are one stretch of sequences. Now here the DNA-A will come and bind. Where will it bind? It will bind onto your 9 mers. So the 9 mers that you see will be here, let's say. So the DNA-A will go and bind here. DNA-A has ATP bound to it, so the ATP will also be attached to it. Just a second. Yeah. So the DNA ATP will be hydrolyzed. Now ATP will take another few more base pairs to sit. So it will go and sit on 4 more nucleotides. So it will actually be a 13 mer, which will be like set upon by the DNA and ATP. Then there will be hydrolysis. And with hydrolysis, you have energy to open up these bonds between the, let's say, between the 8 and 80 of these regions. So with that, you have this first opening of these strands. So you see these strands are being opened up. And then you have accessory proteins like DNA-G and DNA-B. These proteins will be helping to form the replication fork. Now, what is replication fork? What do you mean by fork in general? Fork means something which has 2 prongs or 3 prongs. Here it will be 2 prongs. So once you have this DNA opened up, you have these kind of structures here, replication fork. So that stabilization of these kind of structure, because you see there are 2 single stranded between the double stranded molecule, so they have a lot of tension on their structure. So to release those tension and to actually bind it properly, and to also, now this was opened up by hydrolysis. Now there is something called helicase. Helicase means helix to break the helix. So these are helical structure. So you have some helicases that will open up. Now the bonds will be opened up by these helicases. And where are these helicases? DNA G and DNA B will be responsible for such opening up. So that's what happens when you have to initiate a replication in E. coli. You have first the DNA binding into the 9 mers, ATP hydrolysis, AT bonds breakage, opening of the strand, and then these strands will be stabilized by accessory proteins like DNA G and DNA B. And then that will be followed by the sitting of polymerase so that you can initiate the transcription. So moving on, so as I had mentioned, this is how the replication fork will look. So once you have the unzipping of DNA, this is called replication fork, because at this side you see there are 2 prongs to it. And I had mentioned about helicase. So now this helicase will move in 5' to 3' direction, opening up the strands on this direction so that the DNA polymerase can also move in. So you can imagine, right, as in when these are opening, like for example a zip, if you open it, it starts separating from one another and you have region to where you can move. So for example here, if this opens up, the DNA polymerase will have ample of location to sit and move and replicate the DNA. And primase will come in 2 minutes, but you have a single strand binding proteins and topoisomerase. So single strand binding protein gives stability to the single strands which are formed after the opening up of DNA. And topoisomerase, as I had mentioned, that these structures are under highly structural tension. So these topoisomerases will release those tensions here. Okay, now there is certain problem with DNA polymerase, and DNA polymerase is not capable of synthesizing a new strand. And why is that? Because it does not add nucleotide by itself until it finds a 3' OH free end, and that is one of the problem with DNA polymerase, and because of which you will find that DNA polymerase is not the one that initiates replication. It is rather an RNA primer which initiates a replication. Yes, that sounds a little weird, but that's how it is, because RNA polymerase, or RNA, in this case RNA primase, is capable of synthesizing these strands de novo. So that's term de novo synthesizes. So it doesn't actually require a pre-requirement of a 3' OH free end. It can itself start adding bases according to complementarity. So I'll give you first an idea of how it happens, and then we go step by step. So you have these RNA primers. So these are primer. Why primer? Because they prime the replication reactions further. So that's why it's a primer, and the one enzyme that synthesizes it is called the RNA primase. So the RNA primase will come and sit. Let's say initial 8 to 10 bases it will see from the DNA, and it will replicate, but as an RNA. And now after 10 nucleotides, you have the 3' OH free end, right? So the RNA primase will now move apart, and now the DNA polymerase has enough place to come and sit. And then it will start, because it has 3' OH, it can happily put on new nucleotides after this stretch. But what happens to this RNA? You don't want an RNA in your DNA strand. So later, in this later steps, you will find that all these bases are removed in cell, and then DNA polymerase generally will have a 5' to 3' activity, because that's how replication happens, right? 5' to 3' reduction. But there are some polymerases, or there is also additional activity to polymerase where it is from 3' to 5'. They will move on the opposite direction and try to scan, okay, this is missing, and then it will initiate its 5' to 3' activity to fill the missing part. So that's how your primer is removed and your DNA stretch is added. So now if I take it a little slow, so as I have mentioned, that RNA primer will synthesize the primer here. And what happens after this is DNA polymerase will come and sit. It will have this OH, so it can now synthesize, while moving on the strand, it can synthesize a new strand. Now, how do you remove these bases? Because you see, so if you have to imagine it, the red ones here are RNA nucleotides, and the black ones are DNA. So all these bases will be removed by RNase H. So all the ribonucleotide, not deoxyribonucleotide, but ribonucleotide, because they're RNA, they will be cleaved by RNase H. So the junction between these, so the phosphodiester bond between these bases will be cleaved off. But you have this one last base pair here, which is bound to a deoxyribonucleotide, and it's bound to a ribonucleotide, and this will be cleaved by DNA polymerase 1. So it will be nicking this bond here, and as a result, this all will go up. Now, which enzyme follows it up by filling the gap? This is called something called ligase. Again, as I mentioned last time also, enzymes names are very easy to remember. Ligating means joining two pieces. So here, if you want to put new DNA strand and bind it to the original strand, you will be ligating these two strands. So that's why the enzyme for name is DNA ligase. So DNA polymerase, as I mentioned, that after all this song and dance, the DNA polymerase can finally have this pre-existing free 3' OH, and it can now synthesize the new strand. Okay, so that was all about how you do initiation. Now, how will be elongation? So, elongation means adding of these new bases, right? Now, and this is done by DNA polymerase. But there are several features of DNA polymerase which makes it amenable to elongation and proper accuracy of the steps. So, just a brief idea, we'll do DNA polymerase in a bit. But if you see this region here, this is something like, if you just look at your right hand thumb, it will be something like this. If you do this, you can imagine your DNA polymerase in the cells that you have. So here, the first thing is it will bind perfectly to the template and primer junction. Once it happens, the groove in between this polymer is here, where it is actually bound to these. So you can see this is the new chain which is being synthesized, right? So here, if you see this new base, now this base is adenine. So for it to be very accurate in replicating the DNA, it has to have somehow the mechanism to only allow T, not G, not C, or not A, because A is complementary to T. So what it has is, it has, because this is groove, and A being a biomolecule, or it has its own static properties. Now it has ample of groove that if you have A, the only other base which will fit in the groove is a T. If you have a G, the only other base that will fit in the group is C. And that's how by steric exclusion it keeps up the accuracy of elongation. So just to reiterate, you have these grooves here, and these grooves have only that much distance which will allow the complementary base to attach to the particular nucleotide which is on the strand which is being synthesized. So here, because it is A, the groove will only allow T to come and sit. Because of steric exclusion, the other nucleotides will be not allowed inside the group. And that's how, because T will come closer to A, they will start bonding the two hydrogen, because we have seen this in the last presentation where these guys have, like two donors and two acceptors on their faces, and that's how they bond to each other. That was about how they ensure accuracy. And how does this interaction actually take place? So DNTPs are in its, so the bases in its native form are DNTP. So they are deoxyribonucleotide triphosphate. So they have three phosphates attached to them. But when the phosphodiester bonds take place, in the strand you only have the monophosphate group. So only one phosphate is attached to the base and it remains there. The other two phosphates are given off. So that's what you are seeing here. So if in a strand, let's say you have five monophosphate nucleotides, and now you have a new DNTP added up on the strand. The strand, after the incorporation of the new base, will look like six of these monophosphates, and the two phosphate which was on the incoming base will be released off. So and this is a high energy one. So this phosphate, breaking of phosphate requires high energy, and that enzyme that actually carries this out is called pyrophosphatase. And that energy that you have released from here is what drives the polymerization of these this strand of the newly synthesized strand on the DNA. And this also I have mentioned. Now there is one important factor that you need to come to terms with. So the DNA react, as I told, that the polymerization takes place in 5' to 3' direction. Now before looking at the new strand, just look at the parental strand. So this is one strand, 5' to 3', and this is one strand, 3' to 5', because DNA is anti-parallel in nature. Now you have DNA polymerase on this strand as well, and DNA polymerase on this strand as well. But DNA polymerase can only incorporate in the direction of 5' to 3'. So if your polymerase sits here after your primary synthesis and all that, the only way it can move without any break is this region, because in this region it is the 5' that is being open and up. So when you are actually seeing on the strand, the direction of moving is actually 5' to 3'. So it is a continuous synthesis. And this is called leading strand because it is moving in a continuous way. But now see on the opposite end, what happens? your DNA cannot sit here because 3 prime to 5 prime is not the normal activity of the polymerase. So how does this strand get amplified? So what happens in these strand, which has actually the directionality of 3 prime to 5 prime, there are multiple primers which are synthesized from the direction of 5 prime. So if you have one primer here, this direction is 5 prime to 3 prime. And then so this is being synthesized, this is by DNA polymerase, and this part will be covered by DNA ligase. So you have one strand here. Then after leaving few base pairs, another primer will be synthesized towards its left, and that will also move towards. So like this you have some fragmented amplification. So this is called discontinuous replication, and these fragments are called Okazaki fragments, named after the scientist who actually discovered them. So you have these Okazaki fragments, and the strand itself is called the lagging strand, because the earlier one was leading strand moving in a continuous direction. This lags behind because of the presence of multiple fragments. So that's how the reaction, this replication takes place in these cells. And one more important thing is these fragments also have to be joined to one another. And how does joining happen? Which enzyme takes place of joining in the cells? DNA ligase. So these also will be joined by DNA ligases. Okay. Now, let's come to the enzyme DNA polymerase to understand the structure of the polymerase and how does different part of the polymerase take part in different processes. So, as I had mentioned a few while back, that more or less all these mechanisms are same in the prokaryotes and eukaryotes, but except for finer details. For example, DNA polymerase as an enzyme, there are 3 isoforms of this enzyme, or 3 types of this enzyme in prokaryotes, but there are 5 of this type in eukaryotes. So if you see here, DNA polymerase in prokaryotes are of 3 types: 1, 2, 3, where the 3 is what you generally use for your replication, proofreading, and editing. With proofreading, as I mentioned, there is proofreading activity in the opposite direction, that is by DNA polymerase 3. You have DNA polymerase 1, which is filling up the, so filling if gap removal, DNA repair, and your RNA primers. So the exonuclease activity here is 5 prime to 3 prime and 3 prime to 5 prime. And the second one is DNA repair. So every polymerase basically works by adding nucleotide, but what function it carries forward is why it is classified according to different classes. But if you see eukaryotic polymerases, they are not named as 1, 2, 3, 4, 5. They are rather named as alpha, beta, gamma, delta, and epsilon. And all these, if you see, not every one of them has a proofreading activity. The first one is polymerase alpha, and that has primase. So this is, again, you need primers here as well, because again DNA polymerase itself cannot do. So in DNA polymerase alpha in eukaryote, that's the enzyme that does the primase activity, so it's synthesizing the primer. Then you have polymerase gamma that replicates mitochondrial DNA. So it's very specific that it will only replicate the mitochondrial DNA. Polymerase delta elongates the Okazaki fragments, and polymerase. So you have 2 different type of polymerase replicating 2 different strands in the eukaryotic system. You have delta doing the Okazaki fragments elongation, and epsilon doing the leading strand synthesis. So that's how the systems are different from each other in terms of how DNA polymerase functions. And this is what I was referring to when I had told about imagining polymerase. So just a brief of this. So first of all, you see Taq polymerase, and Taq polymerase is very popular. If you know about the PCR, we'll come to PCR in a bit. But if you know about PCRs, so that's why you find Taq polymerase. How does the name Taq polymerase come? It comes from Thermus aquaticus. And what are the properties of DNA polymerase? Why is it used in PCR? I tell when I come to PCRs. Okay, so if you just put your right hand up like this, you have these grooves, right? This is where your DNA replication takes place by DNA polymerase. So you have these 2 sites, polymerization site and E is the exit site. And in this groove, in this part, you have the strand which is being synthesized. Remember, this is not just like double-stranded DNA here. This is a single-stranded DNA which is unwound first, and then it enters the DNA polymerase groove, and it is being synthesized. So this strand moves here, and the polymerase, so imagine this is the strand. The polymerase keeps moving, sliding on the strand like this, and the new strand will exit from this polymerase at this E side. So this is here, you see that the new strand is exiting from this side, and it is coming out from the polymerase group. And the functioning of DNA polymerase is mediated by Mg2 plus, the cations, the ions that enhance the activity of this enzyme. And this is what I had mentioned, that the geometry of active sites here allow only the correct nucleotide base pair, but sometimes that's how the mutations happen, right? You need some inaccuracy, otherwise it would have, the mutations would have never happened, changes would have never come. But you have certain rate of uh, what do you call it? Like, so that's actually called DNA polymerase fidelity, how much nucleotide it can add in a correct time and how correctly it can add. So generally they will have like one incorrect nucleotide in around 10 to the power 4 or 10 to the power 5 nucleotide. That's very low, but when you compare it across the genome, you see that this matters. And that's how you choose your polymerase when you do your PCRs. Okay, and this I have mentioned that when you have to correct again, for this incorrect incorporation, you do the exonuclease activity. Exonuclease basically means to cleave off the base pairs. And then we have termination. Now, as I mentioned, it cannot go what do you say, infinitely long to just replicate and replicate, because after a certain point of time you will reach another ORC. You can't have two strands continuously being synthesized, because let's say E. coli, it has only 20 minutes to replicate and it needs the entire genome in the new daughter cell. So there will be a regulated process where it will be terminated. And for E. coli, these are called ter sites. So ter sites are termination sites. So what happens here, there are some ter sites on these sequences and you have a fork. So because bubble is opened up when your fork is moving from both sides, moment the fork reaches ter sites, one of the fork is blocked, the other can pass. And because of this torsion problem here, because of this hindrance here, the DNA polymerase is like, it moves apart from the template primer junction, and because there is no polymerase, no extension or no elongation of the strand takes place. So that's what happens here. Do you have an TUS means the proteins that are binding to these sites, and they will be so one of them will be in permissive conversation conformation where it will allow the replication to continue, but another will be locked, and this is where locked will happen. You have this GC which is flipped. So because your polymerase is stopped there, the GC will be flipped out, and because it's a flipped out, it's a like distortion in this stereochemistry. The reaction will be stopped there. Okay, and this is just how DNA proofreading happens. One, it can be because of proofreading, so just moving behind in the 3' 4' 5' direction and checking if actually every gap is filled or not. And the other is mismatch repair. So let's say there is a which has by mistake incorporated G. The A and G will be having a distortion, because A and T are supposed to be stacked to each other. A and G, let's say, cannot stack to each other, because of which the DNA will have a certain distortion, and that distortion is recognized by cell machinery, and then this basis is clipped off and a new base is filled in this nicked portion or the gap. So that's how proofreading and repairs happen. So uh, as I mentioned in the previous parts also, that the mechanism overall is same for prokaryotes and eukaryotes. You just need to remember the basic ways, and there are certain differences as I mentioned about polymerase here. So that's what differs in eukaryote, but the overall procedure is same for both DNA and eukaryotes and prokaryotes. Okay, now we will talk about PCR. And PCR is a technique which is used almost invariably everywhere. Only recently this happened to us, where you had COVID-19, and for you to get tested, that if you are COVID-19 positive or negative, you use the RT-PCR. That means again a PCR based technique to see if you are positive or not. So that's how PCR has gained prominence today in the world. And what does PCR mean? Very simply, if you just expand the mnemonic, it says polymerase chain reaction. So you have a polymerase reaction, but it isn't chains. It's continuous over a certain period of numbers. And you have this DNA fragment which is amplified in thousands or millions of copies. This was developed by Carey Mullis. And the idea here is that there is a short fragment of DNA and which is amplified using primer, and DNA polymerase to give you new strands of DNA, which are again DNA complementary to the template strand. And by the end of your PCR, you will have somewhat like 32 copies. This is just fourth cycle, and a normal PCR is ran for 35 cycles. So you can see how exponentially your DNA template copies are increasing. Why is this important? Because let's say it is important, for example, in RT-PCR for COVID-19, you just required a small sequence to see if you are positive or not, and that will give you an idea if someone carries a particular viral bacteria. It's not just used for a virus, it can be also used for bacteria or any other infectious organism. So PCR broadly has three steps: denaturation, annealing, and elongation. And now, because you have done or you have just gone over the DNA replication, it will be easy for you to understand PCR, how PCR works. So you have your template strand. Now let's say, just like let's take an example that you want to amplify. So you also know that PCR is used in something called phylogenetics, where you understand if today you have seen a sample, a bird sample, and you want to find if this bird is from uh, your particular interesting bird. Let's say it's a crow or is it something else. You want to see by use of amplification of certain marker. But for that, what will you do? You will use your sample's DNA, amplify a certain region, and see to which organism does that match. So what do you use here is DNA to amplify your gene of interest. So the first thing is denaturation, to open up this DNA. So you have this double-stranded DNA. First, they need to be opened up to give you single-stranded DNA. Then you have annealing. Now remember, in DNA application, the first step was primer synthesis. In PCRs, you the like when you are starting your PCR, you give something called primers. These primers are very specific. Now you are giving genomic DNA as an template here, and it has so many sites. If you don't give any site to begin interaction, where will it interaction first? Where will the interaction start? Second, DNA polymerase itself cannot start any new synthesis, so you need to give something that gives them the free 3' wedge. So the primer is very specific to the region of interest that you want to amplify. So let's say this is the gene that you wanted to amplify in your sample. So you have primers in two primers, one primer for one strand and the other for the other strand. Their directions will differ in how they read the sequence, but you have two primers. They are called forward primer and reverse primer based on what kind of strands they synthesize. They're complementary, extremely specific to the sequence that you want to amplify. So that's annealing, and that happens because of temperature. So this one very important thing about PCR, it is thermoregulated. So you put temperatures and you play along with the temperature to initiate, to go ahead through all this process. So in annealing, you will reduce the temperature so that your primers can anneal to these templates. And then you have elongation. Elongation, where you have the DNA polymerase come and sit on this, and then you can amplify these strands. So here, where I was mentioning last time, that your DNA polymerase, which one you are using matters, because some DNA polymerase can amplify or can add 1,000 base pair per minute. Some can amplify very little, with some incorporation of say 900 or 800 base pairs per minute, and some will have very high fidelity. It will stay on the strand and synthesize at least at 1500 nucleotide per minute. Now you want to amplify a certain gene, let's say that is 700 base pair long, and you need to select between your DNA polymerase. So you will select that DNA polymerase that is capable of at least synthesizing this much amount of base pairs per minute. And if you have a shorter DNA, that's okay, because you will be covered in your 1,000 base pair. But if you have a longer stretch of DNA that needs to be amplified, then you need to use respectively the DNA polymerase for amplifying that region of interest. Because otherwise, what will happen is, because your DNA is not very high fidelity, it will move from this strand after a certain time, and your entire length of gene will not be amplified. Okay, so let's try to set up PCR reactions. So today, if you are in the lab and you want to do PCR, there are certain steps that you need to follow. First, you need to have your DNA or your reaction mixture, where you add all these components so that you can initiate DNA PCR reaction. The first thing is DNA template. You need the genomic DNA on which your polymerase will act. Second, the polymerase, the enzyme which will be responsible for amplifying this gene of interest. It can be Taq polymerase, or it can be also PFU. Now, PFU polymerase is better than Taq polymerase because it can, like it is thermostable, and it can adjust to higher temperatures. So when you want to denature DNA, when you want to open up your DNA, you have to give very high temperature so that with the heat, these bonds are broken apart. Now, enzymes or proteins, they are heat sensitive. So what happens if you give a very high temperature and your polymerase only degrades? That's not what you want. So depending on what kind of DNA you have, are they GC reach or AT reach, you will have a certain temperature at which they can anneal or like denature. And you need to use a polymerase which is amenable to this condition so that that polymerase does not break apart. So that's what PFU does. PFU is very thermostable and they do not denature at high temperature. Next, you have this primer. So you add this primers. They are very short. That the best primer sizes are 18 to 25 bases. So you have these short stretches of primers which are in forward and reverse direction. Then you have the DNTPs, the bases in the negative form, so that that can be added by the polymerase. And you have the buffer system where you have magnesium, because DNA polymerase requires magnesium, potassium, which will actually help these reactions to take place. So that's how your PCR mix looks like. There's a buffer, there are DNTPs, forward and reverse, the template DNA, the polymerase, and the water to give you the aqueous environment to carry out these reactions. And this here says, given a particular volume of PCR reaction, how much of each components should be added according to their concentration. So you should always have 1x of the buffer and then certain concentration of the DNTP for all of them to work, and you actually get a product. Okay, now that was about setting up your volume reaction mix. Now putting up reaction. So this is called PCR profile. So the initial denaturation is something is 94 degrees. So what is initial denaturation, which happens for around 2 minutes or 5 minutes? It means opening up of all these strands that you have. So you have and this temperature and this timing depends on what kind of system you're working with. If you have a smaller genome, you take less minutes. If you have a genome which is comparatively higher, then you use larger time for denaturation. Then there are a set of 3 steps which are repeated over and over for 35 times, which are denaturation, annealing, and extension. Now, these denaturation had already happened in the previous step for entire genome. But now, every time you put it into a cycle, the strand that was formed newly for using a primer and your DNA polymerase will also open up. So that's the cyclic step of opening of DNA denaturation. Again, this denaturation is dependent on which gene of interest you are amplifying and how long it is. That's the time and the temperature. Then you have annealing. So now you reduce the temperature so that these opening, whatever opening up it happened has goes down, and you have primers that needs to now align and bind to your specific interest. So you can't put very high temperature because that will not bind. Again, you give ample of time so that the length of the primer can go and bind completely. Then extension, which is where so 72 is the standard temperature at which DNA polymerase adds the new DNTPs, but the time is given by the length of the product that you want. After all this happening for 35 times, you can imagine new strand happening up, you are opening it up again you are doing the same alienating and extension, again you're opening up. So that's how exponentially they will amplify. And then lastly, when you're done with all these cycles, you have a final extension wherein all these remaining ends of the genomic DNA, the new primers or the new strands that are formed, they are amplified. So you give them 5 minutes to cover for all the strands that are there. And then that's how your the cycle for PCR is completed. And lastly, this step hold is where you keep the PCR product. It can be 10 degree, it can be 4 degree. You can immediately also take out the samples just after the PCRs are done. But what it means is you can store your PCR product at like 4 degree or 10 degree for infinitely long. You can pick up your samples after your PCR is done overnight. If you keep them for overnight also, that should not matter. And this is what I was referring to when I was mentioning about the prominence that PCR has gained in the several fields today. First being medicine, where you can test for not only any disease causing organism, but you have some genetic mutations. You can see if any gene is mutated and those that give rise to a particular disease. You can see if you wanted to use gene for a gene therapy, if that gene has been introduced in the particular patient. Second, you have forensic science, where you have these patterning with DNA fingerprinting. So where you can use PCRs, so something like this. Let's say this was from a crime scene, and the crime scene DNA give this kind of pattern, and you have three suspects, and the suspects patterns of DNA amplification is like this. From that you can see which profiles match to the criminal, and that's how you use in forensic science. And then you also do paternity test to understand, because paternity is because of Y chromosome, right? So that's just one chromosome to amplify, so you can check if the Y chromosomes and the Y chromosomes match. uh, and the other is research in genetics. That's what we do. Something like cloning. You can use this for cloning. You can use for phylogenetic analysis, for gene expressions, and there are several other applications of PCRs. Okay. So based on these both topics of DNA application and PCR, there were some questions in the previous year's assignments. So I've compiled them, and I'll read them out and give you 10 seconds to solve them. The first is the concept of central dogma of molecular biology to explain the flow of genetic information. So, who has given this central dogma of life? Then we have primer synthesis in eukaryotes. It's carried out by which of these subunits? If even if you don't remember the subunit, just by the name of it, you should be able to guess. Third is a strand formed after joining the Okazaki fragment by DNA ligase. Is which of the strands? Remember, where do you find the Okazaki fragment? And the last is the proteins that prevent further migration of the replication fork and release the core polymerase in terminating the process in E. coli is which of these? So here, if you remember the like the steps, then you will be able to guess which one is the correct answer. So these are the answers. Crick was easy. As I mentioned that just by looking at the name also you could have guessed. So alpha, beta, gamma are what was the name for eukaryotic systems. DNA ligase, it is with the, like what do you say, with the lead strand and lagging strand, and Okazaki fragments are in the lagging strand. And the fourth one had TER sites and TUS proteins. The proteins, the question is about proteins, so that would be the TUS proteins. Now we have, there are some more questions. The next set of questions are the nucleosome-like structure at 9 mer inducing localized melting at 13 mer is formed by which of these enzymes? Um, Next is the reduction of synthesis of phosphoroester bond during DNA replication is in which direction? The thermostable enzyme used in PCRs, just remember which enzyme we use, where from we use them, and which of these is not a component of PCRs. So the answer for the first one is DNA, and HU means histone proteins. So histone like proteins. So histones are what stabilizes DNA. So DNA A binds to the site that we had already studied. The additional protein that helps DNA is the HU protein. And the reduction of synthesis of phosphoroester bond in the transcription. Just a second, I'm so sorry. In the 5' to 3' direction. Sorry for these confusions. Third one was PFE polymerase. The other one is Taq polymerase. And the component which is not used in PCR is RNA polymerase, because we are synthesizing DNA from DNA strands. Now moving on to transcription, which is one step ahead after you have replicated your DNA. Very generally, this refers to DNA being transcribed to protein, RNA. RNA further is translated to protein. So this is the first step in gene reaction in which information is passed from a gene to the protein. So the ultimate step is protein, but this is the first step towards doing that. And here you have RNA synthesis from DNA, and because it's transcription, the segment that is transcribed is called the transcription unit. And of course, as I had mentioned, that RNA will be translated to protein, so it carries the information that requires to be encoded in a protein. But the difference between DNA replication and RNA transcription is that, as compared to DNA replication, the fidelity or the accuracy of transcription is very, very low. And what does it mean is that the chances of incorporating of mismatches or incorrect nucleotides is more in transcription as compared to the DNA replication process. And another difference between the transcription in eukaryotes and prokaryotes is so I had mentioned that almost all the steps are similar, but there are some finer details. One of the major difference of transcription between prokaryotes and eukaryotes is the way they are transcribed. So in eukaryotes, there is something called monocistronic subtranscription unit, and in prokaryotes, you have polycistronic unit. What does cistron mean? Cistron means is the so cistron is the unit which is transcribed, but cistrons will have its own regions which are promoter and which will help your RNA polymerase to bind. The actual transcription will happen after a certain number of bases, and the actual place where the transcription starts and when it ends is called the transcription unit. But transcription unit with the accessory or additional sequences are called the the cistron. So these cistrons can be either monocistrons, any one cistrons, or polycistron, or multiple cistrons. So in eukaryotes, you have something called monocistron. That means there is only one cistron with one transcription unit. But in prokaryotes, you have multiple cistrons under a single promoter. So if your RNA polymerase sits on this promoter, it can transcribe these different proteins, because one transcription unit leads to one protein. So it can synthesize different proteins by sitting on one promoter. That's why polycistronic. But if in eukaryote it sits on this polymerase sits on this promoter, it will only synthesize one protein, which is called the monocistron. Okay. Now, how does your transcription proceeds? So, similar to DNA replication, here also you have only one strand which is being transcribed. And first of all, it is being unwound. The DNA is unwound, and one strand is being transcribed. But as opposite to DNA replication, the other strand is not touched. It is only one strand that is being transcribed to get to the RNA strand. And where does that happen? Which direction does that takes place? Again, the synthesis from 5 prime to 3 prime direction. So in the original parental strand, so if this is a parental strand, the if it is a 5 prime to 3 prime direction, which other strand is being transcribed? The one which is in the opposite anti-parallel direction. So the 3 prime to 5 prime direction. So here, this strand is being synthesized. So this is called the template strand. Okay. Now template strand and the other strand is called coding strand. Now that is like confusing, right? Why would you say coding strand to the other one which is not being used for transcription? So try to imagine. Let's say this is the region, okay? TCGTAGCA. The other strand has a complementary basis to this. So C, G, T, A, all this. Now in the RNA, also you will have complementary sequence to this strand here. So if you look which one codes for protein, RNA. So when you see strand wise, which is absolutely identical to the RNA in these two parental strands, the one which is here. So to because this one is coding for your protein and this is exact identical sequence as coding strand, because both of them are complementary to this template strand. This strand which is not synthesized or not transcribed is called the coding strand or sense strand, because that gives you sense of the direction of the protein synthesis, while the other one is called the template strand, non-coding strand, or anti-sense strand. Okay. And this is carried by RNA polymerase enzyme, and they are also, because we have seen that RNA also can replicate among themselves, and in those cases you have RNA dependent RNA polymerase, because here you have a DNA as template strand. This is our DNA dependent RNA polymerase. Again, this I had mentioned that all these cellular processes will have three basic steps: initiation to start the reaction or process, elongation to continue and synthesize the desired length of the macromolecule, and then termination to stop the process. So transcription also has these three steps where you have initiation. So the initiation means to actually start the synthesis of the RNA strand. And so for like so your entire genome will not be coded for proteins, right? There are some regions that do not code for protein. They are called non-coding sequences. They're called junk DNA. But then there are some which code for protein. Now your entire thing can get transcribed, but your transcription has to be very careful so that at the stop, when the transcription ends, your protein has that particular stop codon, or your mRNA has a stop codon to stop the protein synthesis. So you have several promoters will actually help you to transcribe these parts. Promoters are region upstream. So what do you mean by upstream and downstream when you talk in DNA strand? Sequences which are to the right of your concerned sequence or focal sequences are called the downstream sequences, and sequences which are to the right or to the left of your sequences are called upstream sequences. So for transcription, you have several upstream promoters, which they will help you to bring the RNA polymerase at that site, and then it will start transcribing the DNA into an RNA strand. And first, the initiation step refers to this bringing of RNA polymerase onto the DNA molecule, and that happens at something called transcription start sites. And then you have, once you have the RNA polymerase sitting there, then you have elongation where it will keep adding these RNA or ribonucleotide phosphates into the strands. And termination is where this elongation or synthesis of the RNA will stop. So, to understand RNA polymerase, you see this is the main enzyme which is involved in transcription. Again, it uses a single strand DNA to synthesize complementary RNA strand in the direction of 5' to 3', and once it reaches terminator, this polymerase will move apart from the strands, and the RNA strand will be released. You can have multiple events of transcription of the same gene. That is called enhanced expression of a gene. So it is not important that one gene is expressed only one time. This RNA polymerase can separate and again come back to bind to this and increase the mRNA level. That's what you do in when you check for your level of our gene expression in these cells. You check how many transcripts have been transcribed for this particular gene, and that happens because RNA polymerase can dissociate and come back and again synthesize a new strand of RNA. Now again, like DNA polymerase, there are some basic differences between RNA polymerases, between prokaryotic and eukaryotic system. So the first is the components of the RNA polymerase. So if you in prokaryotes, you have 4 subunit and 1 sigma factor, which we will see the importance of these factors. And you in eukaryotic, there are 5 types of RNA polymerases, each doing its own activity. Then you have, like I said, there are 5 types, but in prokaryotic, it's only one RNA polymerase with different fact subunits, so that individual activities is done by those different subunits. And so this one enzyme RNA polymerase in prokaryote has 5 subunits, but each of the polymerase in the eukaryotes has around 10 to 20 subunits, which makes them really big sized molecules. And yeah, and one important thing is that in your prokaryotic system, transcription and translation can be coupled. That's called coupled. As and when your RNA is being synthesized, your the RNA can be translated to protein. But in eukaryotic system, there is very elaborate way of how your RNA will be synthesized, how they will be post transcriptionally they will be modified, and then that will be taken to a separate unit to translate. And why does that happen? Any idea? Like, why will there be a coupling of transcription and translation in the prokaryotes? That's because the boundary of nucleus and cytoplasm is very poorly defined in your prokaryotic system. That's why these are coupled with each other. But in eukaryotic system, nucleus is very well separated from the cytoplasm. So your RNA has to be brought to cytoplasm to the ribosome so that you can have translation happening. That's why you have these separate compartments for each of these processes. And there is one RNA polymerase in prokaryote, and we have mentioned, but the eukaryotes have multiple, among which the 1, 2, 3 are what is important for the different type of RNA that we have seen. So we have ribosomal RNA, mRNA, and tRNA. So mRNA is synthesized by RNA polymerase 2, and it is done in the nucleoplasm. rRNA is in the nucleolus, by RNA polymerase 1, and tRNA is again in the nucleoplasm. Nucleolus means absolutely inside the nucleus, nucleoplasm in the surface, in the matrix of the nucleus. tRNA is synthesized by RNA polymerase 3. Okay. So we'll see first in prokaryotes how does the transcription or how does that start site function for prokaryotes. So this is core RNA polymerase. So I had mentioned about 4 subunit and 1 sigma factor. So the subunits are 2 alphas and 1 beta and beta dash. So alpha alpha beta beta dash makes up the core of the RNA polymerase. On this, you have the addition of sigma factor, which makes it a holoenzyme. So what is a holoenzyme? How different is this from a normal enzyme? So the normal enzyme with this, for example, RNA polymerase with these factors or these subunits are not active until it has this factor which makes it an active molecule. So the addition of sigma factor makes RNA polymerase an active enzyme, which is what is called the holoenzyme. Now, as I had mentioned, that there are promoter sites which are upstream of the start site, which will be helping you to bring the RNA polymerase onto the site of transcription. So here, if you see this one, this is the strand that needs to be transcribed. Here, this is the start of the transcription site. Now, 10 base pair upstream of the start site, you have something called Primno box, the sequence of which is very specific, which is TATA-AT or TATA-AT. And then leaving a gap of 15 base pair again upstream, you have something called TTGACA sequence, TT-GACA sequence. The first one is also called Primno box, named after the scientist who developed it or discovered it. Now, you see, what is this minus 35 and what is this minus 10 here? Minus 10, these are also called minus 10 elements. So, minus 10 means minus is a direction. You move to the left, you move down like upstream. So, that is minus. And that is called minus 10, the 10th side, and minus 35, the 35th side. So, you have these sigma factors which come and sit at this side. On the sigma factor, you have the RNA polymerase. So because sigma factor is able to recognize these two sequences, it comes and sits on this side. It also has multiple regions. So the different regions bind to different sites in these promoter element. And then because with RNA, with sigma factor, you have the RNA polymerase coming here. The RNA polymerase will come and sit here. That's the first thing happens. So if you just have to go over the steps in words, so you have close complex formation DNA double-stranded and RNA polymerase bound to one phase of the helix. Open complex strand separated by 14 base pair around the start site. And how does that happen? So this initial site binds to the first nucleotide within open promoter complex at +1 site, which is usually a pure G and A. So after this is closed formation once your RNA polymerase comes and sits. Now once RNA polymerase comes and sits, it will have this start site where you will have the first base as a or a G, and that will interact with the RNA polymerase. And once first base has formed a bond, the other two will follow up. 3 nucleotides will form a bond with the RNA polymerase, makes it stably sit on the DNA strand, and then now what do you need to do? You need to melt the DNA so that you get to the single strand. So here, after this, once your RNA polymerase had sit on the DNA chain, again transcription bubble will happen. So it will use up energy in the form of GTP or ATP to open up this segment of DNA that it is just sitting on, and it will start synthesizing the RNA strand onto the third nucleotide that had been added on the strand. So here, so what happens is every 12 to 14 base pairs are opened up, so that those bonds are broken. There is a bubble formation, and there is a RNA hybrid of 8 to 9, and they keep adding. So 9 base pairs were already there, 3 of them were opened up, so a total of 12 and 14 base pairs have opened up, and you keep adding nucleotides onto the RNA strand. Now there is one important thing that happens here, because this is DNA. Once your DNA has been transcribed, you still need the DNA in your native form, right? So what will happen? When the RNA polymerase is moving along this direction, it will keep unwinding the DNA in the forward direction, but at the behind, when it is done transcribing, it will keep rewinding them. So there is an unwinding and a rewinding happening simultaneously. So that's what we mean by here. So the duplex forms here again, and this RNA, because it is duplex, DNA is forming a bond with DNA, so the RNA is left out. So the RNA is only attached at the one which is being synthesized newly. The other strands which have been previously synthesized are all nascent, or they are outside the ribosome complex. Now this was elongation where you kept adding. Now there is termination. Now termination in prokaryotes are of two types. Something called row dependent or something called row independent. Row is a protein internal to the cell. Now if you use row, or the cell uses row to terminate the transcription, then it is called row dependent mechanism. And if you don't use row, it is called intrinsic. By itself, it is called row independent termination. So first we'll see what happens is row independent termination. So in row independent termination, here is what you see. So in the stretches of DNA, there will be a certain stretch which will be like what do you call them, as complementary to each other, self-complementary and palindrome in nature. So what happens is they form a hairpin loop, and this hairpin loop will be followed by some UU stretches. And remember, RNA is being synthesized as DNA RNA hybrid. So now you have, in the growing chain, you have here formation of a hairpin loop. And then you have some AU bonds between the DNA RNA hybrid. Because of this torsion, a strong structure formed in one single chain, there is a pull, and these AU bonds are weaker as compared to AT bond. So these weak bonds dissociate, and so you can imagine these bonds have broken up here, and these RNAs have now come outside of the complex. So that's independent of row, and uses hairpin structure to terminate the reaction. But if you have row dependent, means you have used row. The row is a 6 subunit ring-like structure, so something like this, a ring-like structure, and it has it binds to the single-stranded RNA. And then it has some sites on the polymerase itself. So it goes up and binds to the polymerase. Once it binds to the polymerase cannot move ahead because it is bound by this protein in its complex. So it cannot move further, and as a result, this has ATP. So again, with the hydrolysis energy, it will move this complex out of the DNA strands or the junction, and that's how you get the RNA separate from the DNA strands. So those are the two steps of termination in prokaryotes. Now moving in eukaryotes, the idea is still the same, that you will need an RNA polymerase to sit on the DNA strand, then initiate the opening of the bubble, and then you will have elongation, and then you will have termination. But how and which of these elements are taking part and what are the names of those elements are where the systems differ. So in prokaryotes, you had Primno box, which is also the Tata AT and TT-GACA sequences. But in transcription in eukaryotes, the elements are called, so this is, let's say this is the initiator. These are some downstream, so you can also have promoter in downstream of the your unit of transcription. And then in the upstream, you have Tata box and TF2B recognition complex. We'll come to what is TF2B in a while. So you have the initiator here, and then you have a Tata box. This is where your RNA polymerase comes and sit. But in eukaryotes, RNA polymerase will require multiple other accessory proteins, which are called transcription factors. The name itself says that they are useful for transcription process. So they will need multiple of these transcription factors to come and help the RNA polymerase in its activity. Now, on this sequence, if you see here, these are very conserved sequences. And what if they are, like for example here, Tata box, these are conserved sequence, but here given the 7 base pairs, these 3 can change, but the last 4 remains constant. Similarly for DPE, generally this is constant, but which of these, so these are how, like A and G can be present at the first site, second will always have a G. So that's how conserved they are. So that's why your RNA polymerase has very specific sites and to recognize and initiate transcription. But it's not only the RNA polymerase which comes and binds. You have these transcription factors which attach or come and bind to this promoter element in a sequential order. So to remember, first you have the transcription factor 2D. So the D will bind, and then you have TBP, transcription factor 2 binding proteins. So you have transcription or so the first sorry. So TBP is Tata box binding protein. So you have TF2D and TBP which come and bind on this Tata box, and then you have a sequence of this transcription factor coming and binding. So after D you have A, you have B, then you follow it up with F, E, and H, and lastly you have J. Now all these different transcription factors have their different activity. One of them will be ATPase, one of them will be helicase, one of them will be topoisomerase, one of them will be stabilizing the structure. So J, the last one, is what is helicase, which will help in unwinding the DNA. And these are therefore called the traversal transcription factor. And BRE therefore is transcription factor to B recognition elements. When it is bound here, this TF2B will go and bind here. That's how this complex is formed. Yeah, like I mentioned that these are basal transcription factors, and so these are unlike of the other promoter elements or start site that we have read, which are AT rich. These are GC rich regions, as you can see from the sequence homology as well. Okay, now once your initiator complex has come and sit onto the DNA, you will have elongation again in the 5' to 3' direction. Similar way, unwinding at the frontal part and rewinding at the rear end. And there will be this bubble formation of every 25 unbound database pairs and addition of or staying of 8 molecules, and the other remaining whatever nucleotide was formed between the DNA and RNA will be broken off, and that will be remaining into the cell environment. And it will, so the RNA polymerase will move along the DNA strand one by one as and when it goes on transcribing each of the nucleotides. And now in termination in eukaryote, so as opposed to what we had seen for the prokaryote, where you had this rho factor either being used or not, in eukaryotes the transcription is terminated by the presence of signals on the or signatures on the RNA or the nascent RNA which is being synthesized. And what are those signals? The signals are poly A tailing. So mRNA in eukaryotes have two kind of modification. One is a 5' capping, and the other is a 3' tailing. So the presence of a poly A tail in the 3' of the RNA gives a signal for the transcription termination. And how does that happen? So you have this RNA being synthesized, and you have this stretch of poly A which has been transcribed. This poly A will be the binding site for several of these proteins, for example CPS7, CSTF. So they will come and they will bind to these sites here, and then they will initiate cleavage. So once that is done, that this nascent RNA which had these poly A will be cleaved off, and that will be used as an mRNA. The strand which was being synthesized on the remaining strand will be then cleaved off and removed from the RNA polymerase and complex. So like that's what I meant. So 3' end processing starts when the cis acting element in the poly A site of the nascent is recognized by the binding factors. And when these factors bind at these end, they form a complex structure, and they result in shear forces because of which the process slows down, and there is a disruption of the polymerase 2 and a cleavage which removes this mRNA from the polymerase complex. Now, as I mentioned, there are two types of modifications, but there are, other than these two also, there are several other modifications that can happen to the mRNA once it has transcribed from the DNA. So these are the several methods. First addition of a cap to the 5' end. Second was addition of a tail to the 3' end. And the third is splicing of the introns. So in eukaryotic mRNA, it is not a contiguous thread that can be translated. You have these stretches of introns, the non-wanted sequences in between two coding sequences. Those are called introns. The one that code are called exons. So these introns needs to be removed so that you have a contiguous stretch of protein. So we'll go one by one over this. So the first is 5' capping of mRNA. So you have the mRNA. Now on this mRNA, you will have this tail being formed here. So the first thing is you add the triphosphate here, a trinucleotide phosphate. and which is generally G in, so a guanosine is being added, and then you remove the phosphates, and ultimately with several modification, the final modified nucleotide which is added onto the 5 prime end of the mRNA is a 7 methyl guanosine. So you had a guanosine, you started with a guanosine, then you added a methyl group on this guanosine at the 7th position, and that is being retained for the mRNA. That is a cap, and it is important because first, it gives stability to the mRNA. Second, it is responsible for export of the mRNA. So mRNA has to be exported to the cytoplasm so that it can undergo translation. So your cap is important with that respect, and and also it is also important for guiding to a certain region of the cell, like for transport and all. Once that is, and the other factor which is important is, so this is a cap to the mRNA, right? So mRNAs or RNAs are susceptible to ribonucleases. So this 5 prime cap protects this mRNA from such ribonucleases. Okay. The second type is the poly-A tailing, which we went over in the previous slide just briefly. So you have these poly-A being synthesized. So with the stretch of the poly-A in the mRNA will be responsible for initiating the cleavage of the nascent mRNA from the complex, and that would be used for your further translation. Now you have splicing. So this is what I was referring to. In eukaryotes, you have these kind of stretches for the mRNA, where you have one exon here, and in between you have this intron region which is not translated. So ideally, if you want to translate a protein from this sequence, you need to remove these introns from this contiguous sequence. Okay, so and your mRNA is so this boundaries of exons and introns where this splicing will take place is very conserved. So every exon and intron boundary is marked by this kind of presence. AG on the exon end, GU on the 5 prime side of the intron, which is called the 5 prime splice site. Then at the 3 prime end, you will have an NCAG. Yeah, sorry for that network connection. All right, so your introns will also have in between a signature sequence where there will be a cu rich region and an a at the branch point. This branch point is important in terms of how they are spliced from the mRNA. So if you just want to overall like get a broad idea of how that happens, so you have these exon ones and the introns here. So you have an so there are two trans esterification reaction which will remove these introns as a lariat and connect the exon 1 and exon 2. So if you see here, this is the stretch of introns between the two exons, and this is first cleaved at the 5 prime side because of the attack from this A on this GNU. So there's a phosphate bond at this junction of exon 1 and intron, and this branch point A first attacks this site because of which this site is cleaved. So now you have a hanging, so this site is open, but this site is hanging with this exon 2. Then again here at this side, the phosphate here, the OH here attacks this phosphate on the 3' side. So here they will attach, and this intron is out as an lariate. So very like to go over once again, you have first this side attacking this side. There's a cleavage here. This part of the intron is moved out, but this is still connected to this site at the 3' splice side with the exon 2. The free 3' OH, which is from the exon 1, will attack this side, 3' splice side. As a result, this part will also get cleaved, and this will be out as a lariate, and this exon 1 and exon 2 will be attached to each other. Now, this is how it works generally. But you have several complexes, several proteins that actually mediate this kind of interaction. So if you see here, you have exon 1 and exon 2. This is the intron, but there are several proteins, for example, which are called U1s and U2 and U3. They sequentially bind to these junctions, and then they catalyze all these attack reactions. All these reactions are catalyzed because of these proteins. Now, introns are also of two types. How they are spliced. So you have group 1 intron. So group 2 is what we read just now. For the A, you have the A binding to the exons, and then they will attack subsequently. So that's how they are lariate in nature, and that's how they come out. But for group, and these are generally the nuclear mRNA, but you have some introns which do not require this A, which is intrinsic in the sequence. They will use G, the OH from a G from outside, and that will be used to initiate all these attacks, all these trans-esterification reactions. So which kind of base that they use for the initial attack, based on that they are classified as group 1 or group 2 introns. And the next thing is, they are not, so if there are, let's say 3 exons or 4 exons, for example, in this case, it's not always necessary that it will be translated in exon 1, exon 2, exon 3, exon 4. It is not a compulsory, because there are several mechanisms which use alternative splicing. What that means is, in some, for example here, let's consider all of these to be 4 exons. In the first case, the third is also, the exon, which is a third exon, is also not included in the final protein. So this final in exons will have 1, 2 and 4, while for the other one you will have 1, 3 and 4. So in one, the third exon is not used, in the other the second exon is not used. And what is the difference, or what is the use of this? In a single unit of transcription, you have this different diversity of protein that can come up. And this is useful in sex determination in Drosophila, where you have these 3 genes which are called sex determination, sex lethal gene, transformer gene, and double sex gene. And because of the alternative splicing, which of these exons are bound to each other, they give rise to either functional genes or non-functional genes in the males and the females. And then you have immunoglobulins. In immunoglobulins, you have 5 kind of immunoglobulins, right? Antibody, IgG-A, IgG-B, IgG-M-E-D. So all these IgGs are formed because of such kind of alternative splicing. So there the genes are called V, G and J, and there are several possible splicing of these 3 genes, and then give rise to different segments of the antibodies. And also hormones. So from one single gene, there can be 2 different hormones or proteins that are produced. For example, the same gene can code for calcium regulating thyroid hormone, hormone in thyroid, but in brain it can give rise to calcitonin gene related peptide. So this is how alternative splicing functions in the mRNA. Okay, now based on these, there are some questions from the previous assignments. So the first question is, the segment transcribed from DNA is called what? So remember, I'll each of these, just read the word and try to understand the mechanism associated with it, and you will understand which one is related to transcription. The second is, the efficiency of the promoter vary with respect to consensus sequence as they vary with the sequence of the promoter. So what it means is, there's a promoter element, right? And there is a consensus sequence. Now, how will the efficiency of this RNA polymerase change with respect to this sequence? A general idea is, if it binds strongly, it will better do the transcription will be more accurate. If it binds just feebly, then the binding accuracy will be less. So based on this idea, let's just try to answer this question. The next is the AT-rich region that undergoes a localized melting in transcription initiation in prokaryotes. So there were two boxes, one in eukaryotes and one in the prokaryotes. Try to remember which one we had discussed. And the next is the enzyme which is used in the synthesis of rRNA is which of these? Okay. So the answer for this is these are follows. So the first is the segment which is transcribed from DNA is a transcription unit because replicon is the unit which is replicated. Amplicon is the unit which is amplified in the PCR reactions. Replication unit is what DNA segment is being replicated. So transcription unit is what is being transcribed. For the second question, the answer is the closer the promoter sequence to the consensus sequence, the greater will be the efficiency of the promoter. Because promoter, as I say, binds to the RNA polymerase, and consensus sequence is what is the conserved sequences across different organisms. So the closer it is to these kind of conserved sequences, the stronger the chances or interaction between the RNA polymerase and the promoter element. So that's why there will be the efficiency of RNA polymerase in transcribing this segment will be higher. And in this, Tata box and Pribno box, Pribno box first for your prokaryote and Tata box first for your eukaryotic system. And for rRNA, we have the RNA polymerase tree. Okay, the next set of questions are the correct order of binding transcription factors during transcription in eukaryotes is which of these order? The transcription factor that binds to the minor groove region of DNA during transcription is which? The multifunctional transcription factor having ATPase, helicase, and kinase activity are which of these following factors? And the enzyme which is not involving 5' capping is which one? So this is the answers. So the first is DABF and then RNA polymerase followed by E, H, and J. So there are minor differences in the sequence order. If you just check the sequences, you can be able to guess which is the correct order. Then you have the translation factor which was the minor group. So the ones which are TAFs and TF2H and these are main factors which bind on the DNA. On the minor groups, you have the accessory proteins. So these are the Tata box binding proteins. Um, in the order of binding, you have H as one of these or the second last transcription factor to bind, and that has the ATPase, the helicase, and kinase activity. ATPase to hydrolysis or energy that gives you to unwind, helicase to break apart the helix, and kinase to move on to the DNA strand for the RNA polymerase. The other one is the enzyme which is not involved in 5' capping. Even though you don't, we did not go over it in detail, but I've spoken about this. That we have this guanosine which is added, then you have a methyl group which is added, and that is added at the seventh position. So all the names here were quite descriptive of the steps that we had discussed. So guanyl transferase, the first addition enzyme which adds the guanyl to the cap. 7-methyl transferase where you have the methyl which is positioned at the seventh position of the guanosine, and 2-O-methyl transferase that adds the methyl group. So there is no process of cycling of the guanyls. So that's why this enzyme is not in the steps of 5' capping. And then the next set of questions are the snRNAs or the ribozymes having the catalytic activity and cleaves phosphodiester bond between splicing is which of these? Basically, which one leads to the actual breaking of the introns and exons? Next is a DNA sequence is read by RNA polymerase that produces complementary anti-parallel RNA strand known as which of these transcripts, which of the transcript that you actually get from DNA transcription, which one is, what is the name for that? And which of these subunits of RNA polymerase is totally required to initiate transcription, which is the factor or which is the one that is responsible for binding? And transcription in eukaryotes is initiated when does that happens? So U1 and U6 are the one that are responsible for the cleavage of the phosphodiester bond. The RNA which is synthesized after transcription is called the primary transcript. Secondary transcripts are the one which are formed after the splicing events and the modifications. Sigma factor, as we had seen, makes the RNA polymerase a holo enzyme other than the cofactor. So that's the responsible for initiating transcription. And transcription in eukaryotes requires the promoter sequence, because without the promoter sequence, you don't have the site where the RNA polymerase will come and bind. All right. So that was about transcription. The final step of, or the final process in central dogma is translation, which refers to the process of synthesis of protein from the RNAs that was transcribed. So, what in this step you have all the three RNAs playing a role. So, mRNA is what codes for the protein. Your transfer RNA, tRNA, what gets the amino acid according to your triplet, codon triplet, and rRNA forms the ribosome, and ribosome is the a place, or ribosome is a site where this protein synthesis takes place. So all the three RNAs that has been synthesized in the cell are used up during the translation process. So here you can see that you have the transcription unit here, DNA replication transcription, but translation is when it moves out to the cytoplasm. This is for eukaryotic system, and you have the ribosome binding onto the mRNA, and it gives rise to the growing polypeptide chain. Again, any such reaction in the cell will always require a energy currency, and for this process, again, you have the use of ATP. Okay, the first thing we talk about is for the how the mRNA is read. Now, mRNA has these sequences. Let's say these are the sequences of mRNA, and how you read an mRNA is called the reading frame. So generally in proteins, the first amino acid is the methionine. So AUG is generally called the start codon. And you have several stop codons for which there are no amino acids. So at those positions, the polypeptide chain stops. So that's where your protein synthesis stops, and your polypeptide chain will stop with the amino acid prior to this stop codon here. So there are three amino acids there are three stop codons, and there is this methionine is generally the first codon to be translated. And how you read your mRNA is called the open reading frame. So an mRNA base generally has 6 open reading frame, which is in the direction of +3 and -3. So +3 and -3. What does that mean is, so let's say this is the reading frame, right? Now, because I know AUG is a starting point, that's why I have said that we'll start reading from this. But mRNA is read in triplets of the bases. So here, because it is AUG from here, this is called the +1 site. But what if your A was here and then it was UG? So let's say there was something called T here. What if you start reading from this? Then you're open, the frame that is actually giving rise to protein is from the second side. So the second amino, if you read from the second amino acid, that is the second frame. And let's say there was another base here, let's say G. In such case, your actual frame or actual side from which the translation starts is at the third position. So that is the +3. Because if you want to move 1 point after, now let's add another base to the left side. Now here, if you add one more base, the frame again starts from AUG because your mRNA is read in three nucleotides at the same time. So if you read 3 nucleotides, the fourth nucleotide is AUG, which is similar to what we read for the first frame as well. So this again becomes a +1 frame. So you have 3 frames in the opposite foreign direction, plus side. And this can also be read from the other side. If you consider this to be a complementary strand and you want to see the actual reading strand, it can also be from the other side. So that left side, it will be -1. If you read from here, it will be -2. It will be, if you read from here, it will be -3. That's how you read the mRNA. Nothing to do with how your directionality happens. But when you read the mRNA, in which direction you read is and which position you start to read, or first which position starts to actually translate to amino acid, is how you determine the ORF. And here, the triplets, each of these triplets give rise to a certain amino acid. So this is the table for that, where you have all the codons and the respective amino acid that they code for. So here, these UAA, UAG and UGA are the stop codons. So these are the 3 stop codons which, if encountered during the translation process, they terminate the translation there. But other than that, if you see, there are 20 amino acids, and there are these many combinations of 64 combination of trinucleotide. So it is very obvious that there is degeneracy. So there is multiple codon that will code for a single amino acid. For example, here, there are 4 codons that code for valine. There are 4 codons here that code for alanine. There are 2 codons that code for serine. There are 2 codons for arginine. And then you have methionine, which is done by one codon only. But other than that, if you move across the table, you see there are multiple codons that code for a single amino acid. And from this, you get another important concept, which is called the Vobel concept. So let's say there are 4 amino acid. There are 4 codons that code for valine, or for example, leucine. There are 6 codons that code for leucine. Now all these, just read these codons. For valine, you have GUU, GUC, GUA, and GUG. So what does it mean? It means that the changes are generally, if you see, all the changes of these 4 codons are at the 3rd position. So you will see in the coming slides that then the tRNA, so I'll just make it for you to understand here. So when you have the tRNA, this is how the tRNA is, and this is how the mRNA is. So these are the mRNA bases, and these are the tRNA bases. So let's say the mRNA base here is G, U, and A. So what does it code for ideally? It codes for valine. Now here, during translation, when there is a bond between tRNA and the mRNA, the bond between the first 2 bases here is very strong, but the 3rd one is not that strong. So if you see these 3 bonds here, the 3rd one is always wobbly, and because this is wobbly and this can't be read properly, so the first 2 amino codons generally, or the first 2 sides of the codon generally determine what kind of amino acid is being incorporated into the growing chain. And that's why the 3rd position is free to change, and that's why you have 4 of these codons to code for valine. Because here the tRNA is the 3rd base doesn't bind very strongly to the mRNA, and that gives rise to something called degeneracy of codons. So you have multiple codons coding for one protein amino acid. So one codon will code for only one amino acid. GUU cannot code for alanine. It will always code for valine, but valine can be coded by 4 codons. This is very important to understand. One codon, one amino acid, or one amino acid can have multiple codons. Okay. So that's what I was referring to. This is tRNA, or what is called the transfer RNA. Why the name transfer RNA? Because this is what is responsible for bringing the amino acids to the growing chain of polypeptide. Means which amino acid should be added into the peptide strand or peptide length is determined by transfer RNA. So structurally, if you see, they are around 73 to 94 in length, 94 nucleotides. And the structure here, so this is how your 5' end is, and then you have this loop here, then another loop here, then a loop here, and this little arm here, and then you have a 3' end. So by name, it is 5' is the phosphate terminal or the acceptor arm. So the acceptor arm in here. So this is 5' phosphate. This is there will be a signature CAA in the 5' to 3' direction. That will be the acceptor, amino acid acceptor. We will see these in the coming slides. What does acceptor means? Then you have this dihydrouridine loop. If you see here, D loop. if you see this uridine are modified to dihydrouridine, then this is anti-codon loop. Why anti-codon? Because it binds to the codon on the mRNA. So it's anti-codon in nature. And then you have this short arm here. And then you have this T psi C. So you have your psi as a base in this arm. So that's a T psi C, or what you call it is pseudo-uridine modified base again. So that's how your tRNA looks. And your CCA is where your amino acids will be bound. And which amino acid binds to this tRNA is responsible or is determined by the bonding between the mRNA and the anticodon. So you have mRNA bound here. If I have to bind, if I have to show an mRNA here, it will be like this. So you have something like this. Yeah. The mRNA is bound here. So you see there are 3 bases here. These will be bound to the 3 bases of the mRNA. This is 5' end to 3' end. So this will be 5' end to 5 will be here. And the 3' will be here. That's how your codon will be read. So if you have to read AUG, the AUG will be like this. And G. That's how. So what happens is you have these amino acids. So you have these amino acids freely lying in your cell environment, and you have the tRNA. Now your tRNA has your amino acid, which charged with the amino acid. So what do you mean by charge it? There are multiple remember tRNA comes with its amino acid bound to itself, but which tRNA gets incorporated into the protein is because of this reaction. But this, if you remove the mRNA from here, then this tRNA will always have one of the amino acid bound to it, and that amino transfer RNA is called a charge transfer RNA because of your amino acid. Now these amino acids can be based on this. So here you have amino acid, this tRNA, and then you have ATP, and then an enzyme that is responsible is called amino acyl tRNA synthetase, and that will add this amino acid to the tRNA, and this tRNA will be called the amino acyl tRNA, and this is what will get incorporated during your elongation. Okay, now the most important thing is there are 30 to 45 different tRNA in prokaryotes and 50 of these in eukaryotes. So the number of tRNA is more than the number of amino acids. So there are multiple transfer RNAs for a single amino acid as well. Similarly to what we read for codon, there are multiple codons for one amino acid. There are multiple transfer RNAs for one single amino acid. And here we also see something called a codon bias. So because there are multiple tRNAs, and let's say you have multiple codons coding for a single amino acid, but one of the type of codons tRNA is more prevalent in the cell or the organism. So for that organism, let's say GUU was coding for valine and GUA is coding for valine, and in some organism you have more of GUU transfer RNA. So there in those organisms, GUU will be it will be more constricted to undergo changes because the tRNA for this codon is more in number, so they have a codon bias. So for valine, they generally use this codon. So this codon is kind of conserved or doesn't get, it's not free to mutate in this kind of organism. So that's called something that refers to something called codon bias. Okay, now that was transfer RNA. Now you have ribosome. Ribosome is where your actual translation takes place. So mRNA is here, and for your mRNA to actually undergo translation, you have the ribosomes binding to it. Each ribosomes in prokaryotes or eukaryotic system, they will have 2 subunits, one large and one small. So for example, in prokaryotic, you will have total of the ribosome is 70S ribosome. S means Sweetberg unit. It means a unit for referring to centrifugation and not as a molecular weight or number. So 70 Sweetberg unit, 70S ribosome. The larger subunit is 50S and the smaller is 30S. And these ribosomes have certain RNA, rRNA, ribosomal RNA. So for example, 30S or smaller one has 16S rRNA, and it's larger, has 5s rRNA and 23 rRNA. While for the eukaryotic, this is the subunits are 60s for the larger and 40s for the shorter. Together, they make up something called 80s ribosome. Larger has 3 rRNAs and the smaller has 1 rRNA, which is 18s rRNA. This is your how your ribosomes look. But if you go further into the structure of ribosome, you will see that they have 3 sites: E, P, A. Exit site, protein or peptide site, or amino acid or acceptor site. So these are the 3 sites which are present in the ribosomes. Okay, so I'll just first tell you a brief about how these processes happen, and then we'll discuss this in detail. So we'll start from here. Again, this also will have initiation, elongation, termination. But the initiation here is very elaborate and very very regulated. The first thing is for these subunits to come and bind to each other. First, the subunit has to form onto the mRNA. So you have first the small subunit will come and bind to the rRNA. Oh, sorry, mRNA, and then you have some factors that, because if you don't need to translate any mRNA, why would you need this big system in the environment? It will keep on going translating every other mRNA. So to regulate this, each subunit, when they are not in the process of translating, they have some factors bound to each at their active site. So they are no more active. So here for initiating translation, you will have these small subunits with their own factors bound to their sites. To initiate the translation, those factors need to be removed, and that will facilitate the binding of the big subunit to the large small subunit. And it is so well what do you say, it is so well structured that at the initial site, when they are blocked here, it will be only one side will be free for the transfer RNA to enter. But once you have the larger subunit attached, you have both these sites to enter for the transfer RNA to enter, and for this to have the polypeptide chain growing. And then finally, when they reach a stop codon, the termination takes place. As soon as termination takes place, this polypeptide chain is released. The ribosomes are released from the binding between ribosome is disrupted, and each subunit again goes back to its nascent form. That's how it takes place generally. And now we'll go in detail about how this happens. So the first step is initiation. Now, again, as I mentioned, the first step is for the small subunit of ribosome to bind to the mRNA. And like I mentioned, such that the initiation codon, that is the first codon, AUG, is in the P site. And how does that happen? So your mRNA in the prokaryotes has something called Shine-Delgado sequence upstream of AUG. AUG is the first codon to be translated, methionine. Upstream to that, there is something called Shine-Delgado sequence, and that binds to the 16S rRNA, which is in the smaller subunit. So it will bind in such a way that your P site, which is the first open site, will be here at AUG, and the first amino acid transfer RNA will enter here. Okay. So your RNA polymerase ribosomes will be structured in such a way that the ribosome goes and bounds to the mRNA, and the P site is on this AUG. That's the first step that will happen. And so before that also, for the small ribosome to bind to the mRNA, there is a factor which is called initiation factor 3, which is bound on this small subunit, which if it is not in function. But when it binds to mRNA, the initiation factor is released, and then you have the first amino acid entering the first amino acid entering the P site, and then you have the elongation step taking place. Now, this Shine-Delgado sequence is very important because it is a conserved sequence at a certain specific site, because it has to be very specific to this binding. Only then will the translation initiate. It is very conserved. It is highly conserved, and you can see that they are complementary, so it is not free to mutate also. That's why it's a conserved sequences. And to go over how this happens, so you have the initiation factors on the subunits, which releases when they bind to the mRNA. Then once you have this binding and the first amino acid entering the P site, the initiation factor releases from the other sides as well, and the large subunit comes and binds to the mRNA. So you have the complete ribosomal setup now. Once your 3 was the first one to get removed so that your small subunit can attach to the mRNA, then the initiation factor 2 is removed so that the larger subunit can come and bind to the mRNA and the small subunit complex. And this happens. Now if you see on this mRNA, there are 3 channels. The first is exit channel. The second is the P site, which has the first amino acid. And the third is the amino acid site or the acceptor site. Now this is the second codon that is to be read, right? So now it has a certain sequence, and based on that sequence, the transfer RNA will be entered. So the transfer RNA has its own anticodon. That anticodon has to bind here. So there are multiple transfer RNA can return enter, but the only one transfer RNA with the specific sequence for this codon, anticodon, will be bound here, and that will then consist of the second amino acid. Now how does that elongate? We'll do in a while. First we'll just check how does that happen in eukaryote. So the same thing happens, the joining of the two subunits in the initiation step, but how they are joined and which factors are joined, they are very diverse in eukaryotes, and that's where these eukaryotes and prokaryotes differ in the way, first of all, the subunits are bound to the transcription factors, and in the way that they scan for the sequence. So instead of shiny Delgado sequence in eukaryotes, there is something called Kozak sequence. That is what is scanned. So in RNA polymerase, the RNA polymerase comes and sits very nicely on the AUG, the first codon. But in eukaryotes, the the ribosome will sit and then scan for your AUG, the first codon, so that it gets to the first translation site. That's the difference between translation initiation in eukaryotes and prokaryotes. Now, for your elongation, what is the idea of protein synthesis? That you have to keep adding to your polypeptide chain. Amino acids has to be entered. So there again is 3 steps. Decoding means first reading the amino acid, first reading the codon. Peptide word formation, getting those amino acid from the transfer RNA to the growing chain, and then the ribosome to move one subunit, one codon ahead. Means you have this sites at starting, right? So you have some codon here, so you have an transfer RNA entering here with its amino acid. This amino acid has to be transferred to this chain, and then this chain will be growing up. The transfer RNA will get empty, and then that transfer RNA will move out. For the second amino acid, the third amino acid in this case, to enter the free site, the ribosome has to move so that it goes to the next codon, and that's how it will continue. So here there are several elongation factors that actually recycle this transfer RNA. So they will get this transfer RNA. They are bound to GTP. So the hydrolysis energy will help them transfer or move across these ribosomes. So, so the proteins that are responsible are called EFTU and EFTS. So you have TU binding to the transfer RNA. They get bind, they release, and then the empty transfer RNA are transferred back to the environment by another elongation factor. So, and eukaryotes use the same mechanism but by different elongation factors. So here the factors were EFTU and EFTS, while here the factors are called eukaryotic elongation factor 1G, 1B. So you see in eukaryotes there is always this requirement of multiple accessory proteins to initiate or to carry forward a reaction, as opposed to prokaryotic, which is comparatively a simpler mechanism. So once you have decoded, then you have to do peptide transfer. So you have this new transfer RNA which has come with its own amino acid. Now here what will happen is, so you have this NC. So from the second amino acid, the NC bond will attack the carbonyl or the COOH end of this chain, the chain on the P side. This is bound, this gets attached to this side. Now what happens? So if I have to just draw and show it to you. So here was a chain and here was a new amino acid. This gets bound to here and this leads to something like this. So this amino acid is now added to the growing chain. This was P site. This was A site. The chain has now transferred to A site. Right? So now when you see after a point of time, you will find the chain on the A site, P site to have a transfer RNA without any amino acid, and E site to be free. So now you have this ribosome to move 3 nucleotide ahead. so that the P site, which had the free ribose transfer RNA, becomes comes at the E site. The A site comes to the P site, and the A site is free again. So again, if I just show you in a mnemonic form, so it was E site, P site, and A site. Now this had your chain, and this had your new amino acid. After a certain point of time, your exit site, your this has now tRNA, but without any amino acid or chain, for example, for that matter, and the amino acid side has the peptide chain. Now this ribosome will move 3 units further. So this ribosome will move somewhere like this, and because of which what will happen is, because it is moving 1 unit ahead, this tRNA will move to E site. The P site will now have this growing chain, and then you have the A site, which is now free to have new amino acid. And once you're free, the new tRNA enters, again the same chain of transfer of the peptide bonds will be happening, and the chain will continue to grow. That's what happens when you have elongation. And this is just to show, okay, again, for this transfer also, you have a translocation, you have factors which are responsible for movement of the ribosome across the codon. And for termination, what is important is first the presence of a stop codon. And when there is stop codon and there is no mRNA, the tRNA that comes into the amino acid side, then the release factors, which are 1 and 2, respective to the codons that they read, they will come in, and then these will undergo GTP hydrolysis to release the ribosomal subunits and release the polypeptide chain. And that's exactly what happens during termination. And in eukaryotes also, the same mechanism happens with the eukaryotic release factor 1 and 3 respectively. They read their stop codon and release the polypeptide chain. So based on all this discussion of translation, there are a set of these questions. We'll try to solve them. The first is, the RNA act as a template and provide the codon information for the incorporation of amino acid. Which of these is the one which is coding for the protein? Second is, the divalent cation required for the formation of ribosomal units and the association of the subunits are which of these? Then the factor preventing the association of the larger than subunit in the prokaryotic system is which of these factors? And the next is, which of these antibiotics inhibit the GTPase activity of the RF3 or release factor 3? So the answers are like this. So mRNA, as we have discussed so far, magnesium is a cation which is very important for any for multiple function processes in the in biological systems. We have seen this in polymerase, DNA polymerase, and now we also see that it is important for ribosomal unit formations. And the third is the first step, where you have the factors that are responsible for preventing the association of 30S and 50S, IF3 and IF2, but IF3 is the first one to actually prevent this binding. And then antibiotic, which is responsible for RF3. Now, can you imagine why they are called antibiotics here? What was the factor, what was the importance of RF3? RF3 had the importance of releasing factor or terminating the translation. Now, if you have an antibiotic, and you want antibiotic means to kill off a cell, so you disrupt this process of regulation and you kill the cell. So all these products that were, or all these antibiotics that were listed here, they work with this kind of phenomena. So for example, trichodermin is synthesized, they are taken from natural sources. For example, trichodermin is from Trichoderma brevicompactum. They inhibit this GTPase activity of RH3, and if there is no RF3 GTPase activity, the protein is not synthesized. The protein is not outside into the environment, and because protein is not there, the important function of the protein doesn't take place. The organism doesn't grow. Like it doesn't grow to the or doesn't function properly. That leads to the death of this organism. Any organism, any infectious, because these are all antibiotics, and because antibiotics, these So all these systems, all these processes are highly conserved. Second that we had read was erythromycin, and I'm sure this would have you would have heard this whenever you had fever or any other problems. So erythromycin is produced from Streptomyces erythraeus, and they inhibit the protein synthesis by binding 50s ribosomal unit. So they bind to 50s ribosomal unit that disrupts the binding for this small and the large subunit. And because you don't have ribosomal subunit, you don't have protein synthesis. You don't have protein synthesis, the organism cannot function. The next one is furomycin, which is derived from Streptomyces albopictus bacterium, again a bacteria, and they inhibit again protein synthesis. So you can see, I had mentioned this protein is so important for any cellular activities. So all these antibiotics work by disrupting some or the other aspect of the protein synthesis. So here, furomycin, they inhibit by ribozyme catalyzed incorporation into the C terminus of elongating nascent chains. So they get incorporated into the polypeptide chain, blocking any other amino acid formation. So there is no proper protein synthesis, and the organism dies. And last one was ricin, which was produced from castor oil plant, and they work by a different mechanism, but again on protein. So unfolded proteins means some proteins which are not properly folded, and in natural system, if you have an unfolded protein or a misfolded protein, they're either properly folded or they are disrupted or broken down, or they are broken up by proteases. But ricin binds to the proteases and do not lead to this activation of unfolded protein response. That means the misfolded or unfolded proteins are not properly folded. So their activity is lost, and as a result, because important proteins are not functioning anymore, the organism dies. So any antibiotic that you use, they all will work on this basic principle of killing of the organism. But and how? By affecting different important cellular mechanism. For example, these are all translation. Like you can have some which are affecting transcription, some which will affect the DNA replication. So the basic idea of antibiotics is to kill off the organism by targeting these important cellular mechanisms. And with that, this session comes to an end. And if you have any questions, you can let me know in the comment section, or you can always post it in the discussion forum. Thank you.